Hello, so we are here today with Jenny VanEyk from the Advanced Clinical Biosystems Research Institute at Cedars Sinai Medical Center in Los Angeles. So, hello Jenny, how are you?
Well, I'm very well. Thank you very much. I'm glad to be here.
Yes, thank you very much for your time. I have some questions for you, starting with your vision for proteomics. So, what are some of the answers in science that proteomics is addressing, and how is this contributing to life for humanity?
So that's a really big question.
Yes, it is. It is.
So the new research institute that we've just started at Cedars Sinai is really trying to address some of these issues. So the concept behind it is to be able to bring discovery, really proteomics discovery, all the way through to patient care. And that means we need to be able to impact healthcare and healthcare delivery. And one of the ideas is to be able to make an efficient pipeline that can allow us to do very good protein quantitation at discovery, have it through the replication and validation, whether or not we're chasing down biomarkers for a specific disease entity, or whether or not we're trying to reduce the number of side effects where we're doing screening for drugs, and take that finally to a product that we can actually put into the clinic and use it. And Cedars Sinai is a very good place for translational research.
Okay, thank you. My second question is, how has the field of proteomics evolved and where is it going?
So I think this is a very magical time in proteomics. For a long time we've promised many things that we could be able to do. And I think at this point with sample prep and with the mass spectrometry, especially with the new technologies around swath and swath-like technologies, coupled with the really high precision quantitation that we see in the triple quads and the 6500, that we're at this moment that we can make this leap. And the leap will go to higher throughput. And as soon as you can do higher throughput, you can start to do many, many patients, but you can also start to do many more experimental data sets. So you can take more cells, more cell conditions that you're going to be doing, more times of drugs, more times of delivery of that drug, and to be able to get really this dynamics and information that we could never see before. And when you layer up all the post-translation modifications that have been, the tools have been developed over the last few years, now you can start to see that we can just begin to really grasp what's going on at the proteomic level, and then harnessing that through to patient care is, I think, the next step.
Okay, thank you. And my last question for you is, how has AB SCIEX technologies contributed to your research objectives?
So a lot, actually. So AB SCIEX, we first started to work with them really on the triple quads when we wanted very precise measurements of proteins and, well, the peptides that represent those proteins. But because we worked so much on post-translation modification, it became critical that we had the high sensitivity and reproducibility, because we need to be able to determine site occupancy at the actual amino acid that's modified. And this allowed us to do it. And also because we work in the clinical area, move towards large sample numbers, we need the reproducibility along with the sensitivity. And certainly the 6500 has done very, very well for us. When SWAS began, we were very excited and remain very excited about it. The technology has really allowed us to get that same level of quantitation, but on a broader scale, and be able to really start to look at it for tracking mechanism, to be able to track different post-translation modification, and then once we understand enough, flip that over to be able to use the triple quads for these very large numbers that we're using in our cohorts. So I think the instruments that we've been using have been incredibly helpful for us.
Okay. Thank you very much, Jenny, for your time. Now, if it's okay, we can drink some coffee. Thank you. Enjoy the conference, and you stay updated.